A good day to everyone. Today, my groupmates and I will be talking about a few things. But first, let us talk about the do's and don'ts in a laboratory.
First off, we should dress appropriately. Long pants and shirts that cover the whole torso are strongly advised, as well as shoes that cover your whole foot so that when you drop something, your feet are safe and sound. Long hair should be tied since it can get in the way of a chemical solution or open flame. You are also not allowed to wear contact lenses since chemical vapor may penetrate the lens, which may cause the lens to adhere. And of course, always wear your lab gown. Do not eat or drink inside a lab since you can contaminate your samples, and no one wants to start an experiment with a mistake that can be easily avoided. Other things that you should always remember are: first, keep your work area neat and free of unnecessary objects. Never pour chemical waste into sink drains or waste baskets and place them in appropriately labeled waste materials. Never point the open end of a test tube containing substance at yourself or others. Label your samples properly so that further in the experiment you won't get things mixed up. Inspect your equipment for damage prior to use and always wash your materials after the experiment.
A measurement is a collection of quantitative or numerical data that describes a property of an object or event. It is made by comparing a quantity with a standard unit. Since this comparison cannot be perfect, measurements naturally include error, which is how much a measured value deviates from the true value. There are 7 base units in the International System of Units: the kilogram for mass, second for time, Kelvin for temperature, ampere for electric current, mole for amount of substance, candela for luminous intensity, and meter for distance. Data are individual pieces of factual information recorded and used for the purpose of analysis. It is the raw information from which statistics are created. Statistics is a branch of mathematics dealing with the collection, analysis, presentation, interpretation, and conclusion of data. Statistical treatment is when you apply a statistical method to a dataset, which can be either descriptive statistics, which describes the relationship between the variables in a population, or inferential statistics, which test a hypothesis by making inferences from a collected data. In descriptive statistics, there is no uncertainty. The statistics precisely describe the data that you collected. If you collect data from an entire population, you can directly compare this descriptive statistics to those from other populations. This statistics characterizes of your data by the distribution, which concerns the frequency of each value, the central tendency on the averages of the values, and the variability on how spread out the values are. Inferential statistics have 2 main uses: making estimates about populations and testing hypotheses to draw conclusions about these populations. With inferential statistics, it's important to use random and unbiased sampling methods. If your sample isn't representative of your population, then you can't make valid statistical inferences.
Hola! Many of you are familiar with concentration and diffusion, right? They're like famous celebrities when it comes to data calculation. Today, I'm going to show you how to calculate them in easy peasy steps. Concentration is the quantity of solute in a given solution. There are 2 ways to calculate concentration. First, through simply dividing the amount of solute by the total volume of the solution. Second, for instrument-based experiments, the construction of the calibration curve. On the other hand, dilution is the addition of solvent to a solution to decrease its concentration. The volume of solvent needed to prepare the desired concentration of a new diluted solution can be calculated using the formula M1V1 equals M2V2. Calibration curve is a plot of instrument response to analyte concentration. Using spreadsheet, first, identify your X and Y values. X is usually the concentration of the known solutions, while Y is either absorbance or intensity depending on the instrument used. Next, select all the data and then go to Insert tab. In the Insert tab, choose Scatter Plot, and then there you have it, your linear calibration plot. You can solve for the concentration of unknown solutions by calculating the X value in the regression equation given from the chart elements.
In laboratory, pipettes and pipettors are fundamental tools used to transfer and dispense fixed amounts of liquids. Pipettes come in several designs for various purposes with differing levels of accuracy. Volumetric pipettes are designed to deliver accurately fixed volume of liquid, whereas measuring pipettes can measure a range of volumes. More pipettes and serological pipettes are measuring pipettes. Calibration markings on more pipettes begin above the tip, whereas serological pipettes are calibrated to the end of the tip. Multi-channel pipettes generally come with either 8 or 12 pipette heads, allowing to fill multiple wells at a single time. Micropipettes are utilized to transfer small quantities of liquid, usually down to 0.1 microliter. Now let us learn the basics of pipetting. There are three positions: the ready position, the first stop, and the second stop, and an ejector button. Pipetting begins as the tip is attached. Apply light pressure to insert the pipette into the tip. The aspiration angle should be vertical. Press the control button down to the first stop and immerse the tip into the liquid. Pre-wet the tip 2 to 3 times, then dispense all liquid before aspirating. When dispensing, the angle of the pipette should be between 20 to 45 degrees with the tip in contact with the vessel wall. Press the control button down to the first stop, then perform the blowout by pressing down to the second stop. Lastly, press the ejector button to eject the tip.
Now, let's apply what you've learned so far by performing a buffer experiment. A buffer is a solution that can resist changes to pH when limited amounts of acid or base are added to it. Buffers are created by mixing a weak acid or base with its conjugate. Because both the acid and the conjugate base are present in such a solution, they are able to neutralize small amounts of any acid or base that is added to it. Several biological fluids are buffer solutions because they must maintain a steady pH to avoid loss of biologically important functions. The bloodstream, for example, must maintain a pH of roughly 7.4 in order to deliver oxygen throughout the body, and if it changes radically, the proteins in our blood start to denature.
Now let us talk about a procedure for making buffers. First, we measure out 30 ml of water into two separate 250 ml Erlenmeyer flasks, and then we record the pH of the water. Add 2 ml of 1 molar hydrochloric acid to one flask and record the pH. Add 2 ml of 1 molar sodium hydroxide to the other flask and record the pH. We label the flasks to keep track of the solutions. The table will serve as a guide to prepare for the buffer solutions. We record the initial pH of the buffers A to E. So for each buffer, there will be one where acid is added and one where the base is added. We then add 2 ml of 1 molar hydrochloric acid to each of the buffer flasks that are labeled plus acid and record the pH. Similarly, add 2 ml of 1 molar sodium hydroxide to each buffer flasks that are labeled as plus base and record the pH. We then calculate the theoretical initial pH of solutions A and E. Then, calculate the theoretical pH of solutions A and E after 2 ml of 1 molar hydrochloric acid was added.
Now let's proceed to the data calculations. First is to list all the given values and the evaluation which shows that the change in concentration of the reagent has an effect on its pH. The next calculation is for the pH of buffer A. Then the pH calculation for buffer B. Next is for the pH of buffer C. And the pH calculation for buffer D. Lastly is for the pH of buffer E. As for the small addition of strong acid and strong base, here is the pH changes for buffer A, here is for the buffer D, and finally here is for buffer E.
In this experiment, different types of buffers were prepared to understand the relationship between its composition and its behavior towards the addition of acid or base. Here are some buffers prepared with its corresponding pH. Stock solution in flask 1 composed of 30 ml water and 2 ml of 1 molar HCl had a pH of 1.2, while flask 2 composed of 30 ml water and 2 ml of 1 molar NaOH had a basic pH of 12.79. Meanwhile, water has a neutral pH of 7.
Buffer T had the most acidic pH of 4.05 since it was composed mainly of acetic acid, while buffer A and B had the same pH of 4.72. Buffer C and E also had the same amount of pH, 4.76, which was the largest among the 5 buffers prepared.
In conclusion, the acid-base composition of a buffer has an effect on the behavior of buffer solutions in terms of pH value. Any addition of strong acid such as HCl or strong base such as NaOH results to a small difference in the final pH value of a buffer.